epileptic patients who are having something done anyway, if we're trying to localize a source or what might be the start of a seizure, in some cases you can get these patients to agree to have electrical recordings done while you're doing the surgery. So there is, interestingly, that data. So there's much more. The main message of this is that there's much more than these population recordings that are external to the body. You can look at extracellular activity, which means you have a wire or some other type of electrode in the nerve tissue, in the brain tissue rather, and it's near neurons, and the neurons are spiking. And you can pick up on it. How many people know what a neuron is, or at least have had sketches of explanations? Yeah, sketches of explanations, maybe. Well, the basic idea, I'm going to touch on it for those who don't know. Your nervous system has a lot of neurons. It also has other things in it called glial cells that support the neurons and other stuff. But neurons we tend to think that neurons are the seat of computation. If we're ever going to find consciousness or other magical words like that, there will be somewhere in the neurons in their structure. Neurons are typically organized by a soma, a body, which has the nucleus, classic cell structure, animal cell structure. Dendrites. So in the top left of this figure, you can see a lot of things webbing out. And those are called the dendrites. They receive input from other neurons. And again, I'm speaking at a high level. It turns out it's very hard to be much more precise than this. Neuroscience is a fairly young field. But there's some kind of integration happening, as in the neuron picks up this activity from other neurons in its dendrites. If you look in this figure, the soma has a purplish color. Now, this isn't made to be biologically correct. It's just done, the color scheme is done to be more clear. Attached to the dendrites, you can see these bluish purple things. Those are axons from other neurons. And so this neuron has an axon that comes out. It's covered by that orange material. And what that does is it speeds up transmission. It makes the transmission mostly electrical. We can talk more about that in a moment. And then this axon goes on, and it goes on to touch other neurons. And there's things called synapses. Maybe you've heard about them. When you do drugs, typically it's in this synapse where activity happens. So there's chemical transmission between an axon of one neuron, dendrite of another, and selective serotonin reuptake inhibitors, antidepressants, that's a class of antidepressants, selectively inhibit the uptake of serotonin in these synapses, and then the effect is people tend to be happier with other side effects. And so that's a sketch of the neuron. I showed you this, and I spent just a moment on it to try to convince you that there's potentially lots of richness here. We know the richness because that's when you have a drug or when there's something affecting your nervous system, it's happening at this level, at the neuron level. And so if we're going to try to understand the nervous system or biological systems, we need to go down to a fairly basic unit. I propose it's the neuron or something close. You can also do something called intracellular recordings. That's where you actually have a pipette or an electrode where you stick it on the surface of the neuron and you pull off some of the membrane. And then you can actually record intra-cell, inside the cell. So it's extremely difficult to do, but you can get extremely good recordings. They're good because I didn't mention this before. And again, please interrupt me when things are clear or seem understated. All this integration that's happening at the dendrites, supposedly, is encoded in spikes. So there's some threshold. And when the potential across the membrane goes above some threshold, the neuron spikes. And when you stick an electrode, so if you caught a fruit fly, if you let out a banana in this conference and then came back tomorrow and got the fruit flies, you could actually record from them. And if you were recording their neural activity, you would see spikes, extracellular. That's if you stick an electrode near a neuron, the strongest electrical activity is the spiking. If you do intracellular recording, then you can actually pick up what's called subthreshold activity, sub the threshold of spiking. And you can't do any of this with non-invasive imaging techniques. So fMRI, you're Outside the brain, you're picking up gross levels of activity. We could talk more about that if you're interested during questions, or now. So a bat brain is very small. Bats are small, their brains are smaller. The skull bar on this plot is 5 millimeters. We're looking at the top view of a bat's brain. So its mouth would be on the left side of this figure. Its back and body would be to the right. And I show you this just to give you, well, we're not going to draw much benefit from this aside from you seeing the gross structure. There's the SC and the IC. Those are the superior colliculus and the inferior colliculus. As mammals, we have them too, but in humans, we have a huge cortex, and it sits on top of the brainstem and other brain areas. But in bats, conveniently, there's not as much cortex, and the IC and the SC are exposed. I mention those because they have fairly interesting behavior. For example, you can find neurons in IC that are what are called echo delay tuned. As in, if you have a bat and you have an electrode in its IC at some level, and you play to the bat a vocalization, a pre-recorded vocalization even, one of those that I showed you earlier, you'll see some, well, you won't see activity actually. You might see activity for certain types, but for echo delay tuning, or echo delay tuned neurons, you won't see the activity until you have an echo at a particular delay. And in fact, these neurons will fire or spike more often depending on the delay. And this is interesting because already we see some manifestation of range measurement. As in, if you had a population of these neurons that are delayed to echo, tuned to echo delay, you could find out or measure which ones are firing and get some sense of distance. Is that clear, that basic process? And you can also find neurons that are selective to azimuth and elevation, that is direction. And so when you combine them all, you already have a spatial view, acoustic spatial view. So one moment, look at the time, okay. So the bat's vocalizing. You saw a time waveform, you looked at a spectrogram, but that isn't enough. If I had a speaker in front of you, typically it would have a piston operation. We could think of it as a piston being a cylinder that moves like this. It moves the air in a particular way, and you can model it fairly well. But humans and bats and other animals don't have such a trivial shape to the way they generate sound, which is why it might be interesting to study the sonar beam. I say it might because we don't know, and I'll touch on some work here. But the sonar beam refers to if we took a picture, so if we picked a particular frequency and a particular time, we could get some idea of shape of what the bat emitted. So previous work, so it's a difficult problem because bats are flying. Animals, and if they're not flying, or if they're what is called head fixed, as in the head is fixed in place by something that's cemented to the skull, the bats don't want to do anything. Maybe that's not surprising. The closest we can get to a bat not flying and vocalizing is what are called platform experiments, where you train the bat to remain on a platform and do something, vocalize out in a space, make a decision. For example, there's a whole paradigm of these experiments called two-alternative forced choice, where the animal is forced to make a choice. Forced in the sense that if it gets off the platform, then that doesn't count as a trial, but it must indicate, for example, from this direction, the delay was shorter than this direction. Okay. Yeah. And so earlier work, beam shape of elicited calls that I wrote here. In a head-fixed animal, these people, or these scientists, whatever, stimulated the brainstem of the bats and were able to elicit vocalizations. The problem with this study is those are synthetic in the sense that the bat wasn't vocalizing naturally. It was vocalizing because someone was putting current through some part of its brainstem and didn't really know what was going on. It just so happens the vocalizations come out. So later work is with bats that are freely flying. It doesn't include any neural work because currently the electronics don't exist to record from a bat that's flying freely, the electronics for the neural system. There's some work to do that, but it isn't widespread yet, as in it's just in some lab and they're not really sharing it. Not with bats, with another animal. And so, anyway, this earlier work, so for example, in an array I'll talk about in a moment, a previous researcher found that there are two lobes. So if I put a piston in space and I'm emitting a tone from it, it has a typical structure of one big lobe, and this is what you would see with ultrasonic rangefinder, for example, simple sonar devices that have this piston shape. And there'll be side lobes, but they're small. But this person found for the bat, when it's vocalizing, it has the forward beam. These were fairly gross measurements. There wasn't much precision in the shape, but there's a gross beam, and there's one that comes downward. So from a side view, that means when I vocalize out, there's a primary beam, and there's one below, aimed downward. And so then we might ask, why does the bat have that? And the simple answer is we don't know. One idea might be that the bat can use it when it vocalizes to both vocalize ahead for tracking insects or targets, and it can use the slower sound to get more energy to come from the floor or the ground to get a sense of altitude. And there's other work involving tracking. So when you have the arrays set up that we'll talk about in a moment, you can look where the most energy is directed in the vocalizations and get a sense of where the bat's looking. Again, that'll be more clear in a moment. And I'll skip the last off-axis aim. I'll go back to it. I don't think it's so clear what I'm saying without pictures. But the pictures will come soon. So microphone arrays. If we want to measure a sound, if you have a point in space that is a microphone, you're not going to get any sense of shape. And so there's this thing called a microphone array, which is an array of microphones, as in you're sampling the sound field in space. The previous work to do that with freely flying bats was an array that just had a fairly simple electronics. The basic idea is a band-pass filter centered at 35 kilohertz. It's fairly tight, which means you're primarily seeing energy at 35 kilohertz. And so in case anyone didn't know, human hearing is up to about 20 kilohertz. It goes down as you get older. And so with this work, or with that array, which is one of the first, if not the first, for bats, that's how this researcher showed that there are these two lobes, and you're able to study tracking now, because you get, when you construct a horizontal array, so if I'm sketching out a room and you're looking down on it, a horizontal array means that microphones go around the room. And when a bat's flying around the room, you can see which direction the bat's vocalizing. And I'm going to skip ahead to show you the video just to keep everyone interested. Let's start it over. Or not. Let's wait till the end and then go again. So there's a bat moving through this field. We're looking down on a room. The large circles with the black dots in the center are those artificial trees that I showed you before. The magenta line is that net that prevents the bat from circling around the forest. Since we're looking down on it, it just looks like a line, but again, it's a sheet. And the blue circles that are around the perimeter are the microphones in this array. And so the blue trace, if you didn't guess it already, is the bat's flight path. The blue circle is where the bat is at the moment. This video is shown at a rate that's a factor of 10 lower than what actually happened. Good. The greenish diamond is where the microphone is on the floor that's used as a reference. So the array only picks up energy near 35 kilohertz. You don't get actually a full shape of a call with time. The green microphone, or the green point, shows where a microphone is that would show you where you could pick up a full band. As in when I showed you an example call earlier, that was a wideband recording. There's no filtering to happen. Well, there's filtering to prevent something called aliasing, but yeah. Is that good? What units? Time. So 1 second in this recording corresponds to 100 milliseconds. It is the actual sound. And so when you slow it down by a factor of 10, then all the frequencies effectively shift down. You can see this effect if you play back a tape at a higher rate, everyone sounds like chipmunks. If you play it back slower, people sound like something deep.